Wir haben nun noch einen Vortrag aus einer ganz anderen Ecke der Informatik. Gene Myers wird sprechen über die Rolle der Informatik in der Biowissenschaft. Er wird über Vergangenheit, Gegenwart und Zukunft reden. Wir freuen uns, dass Gene Myers uns heute den Vortrag zugesagt hat. Er ist ja einer der bekanntesten Forscher in Deutschland, Direktor des Max-Planck-Instituts für molekulare Zellbiologie und Genetik und Leiter des Zentrums für Systembiologie hier in Dresden. Ich bitte Sie, ein bisschen Ruhe zu bewahren beim Umorganisieren, weil noch die anderen Standorte zuschauen. Es wird noch live gestreamt in die anderen Standorte. Ja, Professor Myers, Sie waren einer der Wissenschaftler des Human Genome Projekts in den USA und sind jetzt hier in Dresden mit der Forschung von Wachstumsprozessen von Lebewesen und Organen beschäftigt. Ich habe schon einiges an Informationen, Filmen und Vorträgen gehört, dass da signifikante Fortschritte zum Beispiel beim Wachstum von Organen erwartet werden und auch erforscht worden sind schon. Deswegen bin ich sehr gespannt, wie Sie uns zeigen werden, wie für solche doch sehr essentielle Probleme des Menschen, wenn man älter wird, spürt man, man muss seinen Körper ab und zu mal in die Reparatur geben, in Boxenstopp, wie da die Informatik helfen kann durch Simulation, Visualisierung und virtuelle Realität. Wir freuen uns daher sehr auf Ihren Vortrag. Bitte schön, Professor Myers.
Can you hear me? Yes. Can you hear me? Yes, it's good? Okay, thank you. I'm honored to be here, although a little bit disappointed to see that biology is less interesting than robotics. Forgive me for speaking in English, but I still can't manage an entire talk in German. So I hope to entertain you just briefly with a perspective about the role of computer science in biology and in the sciences in general. So in the past, I kind of break time into two big epochs. In the initial days, back when computer science was first starting, in the 50s to 80s, when also actually molecular biology was starting in 53 with the discovery of DNA. Most of the work was focused on basically trying to understand the large proteins, the large molecules that affected life and their structure. And then in the period, really in 1980 to 2000 is the period when computer science began to enter, because that was the period in which we began to be able to actually sequence the genomes of a variety of organisms. And so suddenly we had interesting data sets. So again, to basically, back in the 50s, the early work was basically on trying to determine the structure. Computers were used in very simple ways to augment these calculations, and there was a focus on doing large proteins. In fact, it wasn't until 2013 that this work was acknowledged. And so this is interesting, this Nobel in 2013, these three men here, Karplus, Levitt, and Varshall, basically all did computational work. Their work was basically on how to simulate large molecules, and that was finally recognized in 2013. What happened in basically the late 70s was that several people, Wally Gilbert, Fred Sanger, and a few others, basically began to develop techniques for automatically sequencing DNA and sequencing proteins. And the Nobel was given somewhat a more timely fashion in 1980 for this. And at that point, basically DNA sequencing took off, and with it, basically the area of what we now call bioinformatics really began. So bioinformatics really didn't start until the sequence data was available. And the interesting thing that I think about that is it was a real collaboration between computer scientists and the biologists. And if I could, the reason was that biologists are primarily not trained analytically like physicists and chemists. They have to learn a tremendous number of things about how these complex systems work. And so they spend most of their time trying to understand and learn what's known about how these systems operate. And so they're not really able to manage complex computations on their own. And so they needed help. And the computer scientists, of course, and then the computer scientists, on the other hand, well, we don't know a thing about biology. We did understand strings, we understood text. In fact, many of the earlier techniques that we worked on for strings are now used by Google and others for search. So search was a problem we understood. You know, I've never had a physicist or a chemist approach me and ask me how to solve a problem. Never, okay? Because they think they can do it. The only time I ever had a request was they wanted me to make it faster. Okay, they do recognize that we make things go faster, but they think they know how to compute as well as we do. So in that period, there were a couple of killer apps. One was the BLAST program, which was a program for basically searching the DNA sequences, and another one was ClustalW by my good friend Des Higgins, which was a tool for basically aligning DNA sequences. And these papers have a tremendous number of citations, okay? So if you want to have an impact as a computer scientist, do one of these. In fact, I remember in 1993, I was in a very small department, computer science department, 12 faculty, and suddenly our department was number 3 in the United States in citations per faculty member. This right here, okay? So suddenly, I mean, so think about it, okay? I mean, just an idea, okay? The other, I think, really significant thing in that period was the sequencing of the human genome. And as you know, there were a couple of papers. There was a public effort and a private effort. And you may also have understood that there was it was a bit of a competition, and a number of books have been written about that competition. I mean, basically what happened is in the mid-1990s, Jim Weber and I proposed that instead of organizing, so the current technique was to basically subdivide the human genome into 50,000 kind of big pieces and understand where those pieces were. And then they were going to basically do the DNA sequencing on each of those big pieces. And they effectively had a 1,000-piece jigsaw puzzle. And what Jim and I said is, no. If you collect the right information, just do it as a 50 million piece jigsaw puzzle. Just do it as one big jigsaw puzzle, and it will save you a tremendous amount of time because you don't have to understand where to put those 50,000 pieces. And I thought, I actually toured Europe trying to get people to promote this idea. Basically, the powers that be in the public sector said no. But in 1998, Celera was formed with the idea of doing exactly this. It's jump-started. So I basically went. I had a crazy little crew. And you see, the thing that's interesting that I think is that it was the first time that there was a problem, a computational problem, this 50 million piece jigsaw puzzle, where basically the scientists, the non-computer scientists, all went, no way, you're not gonna be able to do this, okay? But they were, fortunately for me at least, they were wrong. Okay, which is that, yes, it was possible, and I don't have time to explain exactly how we solved the technical problems, but I had basically used simulation and a number of techniques to understand that from an information perspective, there was sufficient information to do this. Of course, when I went to the company, I didn't know how to do it. That was still a problem, but I did manage it. And I think what's important for computer science is that basically that informatics, this computer science, provided a pivotal role. It provided not only the technique, the idea of the approach, but it actually was the first time where I think that the scientists saw that computer scientists can do something that they can't do. You know, a lot of people, biologists, they just want to basically transform their data. They want to do what they're doing. So if you can give them, they're happy with what I call the 75% solution, right? If somebody not particularly expert in the skill kind of, can do this, right, then they're happy, right? But they don't really appreciate that they have the 75% solution. So if you think about a lot of the problems that occur in the real world, there aren't that many where people go, no way, okay? And we actually, so I was very happy for the field that we were perceived as making a contribution that surprised everyone. Okay, so today, in the present, basically, after sequencing the human genome, basically the goal became very medical, and the focus was to reduce the cost so that it costs $1,000, 1,000 euros to resequence a person, to basically understand how Professor Nagel is different from me, okay? And then there was lots of work on basically the genotype phenotype. In other words, we look at different people's genomes, we look at the differences in their genomes, and then we look at how they present physically. Do they get a heart attack? Do they die early? Do they get Alzheimer's? What's going on? And by simply looking at the correlation, trying to understand those things. And there was also lots of work on trying to understand how cancer was scrambling genomes, as a hope that would reveal something. So honestly, from my perspective though, because I'm a scientist, I'm interested in science, I was very disappointed because basically after that point, there has not been, okay, there was a 15-year period from 2003 to 2016, a 13-year period, where not one new high-quality genome was produced of something other than, so we had the genomes of human, mouse, gorilla, fly, worm, yeast, and lots of bacteria. And we were sequencing other things, kind of understanding their proteins, but we were not producing genomes. And because we were basically focusing on technologies that were aimed at that $1,000 figure. But it did do, but having these genomes of these model organisms really did transform and aid biology. So it was not a waste of money, in the sense that basically, typically inside of a genome, if you think of this as a long sequence, and I'm just showing a part of it, basically there's a region that controls the production of a gene, and that gene is converted into a protein in the cell. So by transgenic means, we're now able to basically take this part of the genome, but you have to have the sequence in order to do this, okay? That's the important thing. You can't do transgenics unless you have the sequence of the genome. So suddenly for mice, for flies, for worms, for interesting organisms, we can do this trick, which is we can basically add a fluorescent protein, a coding for a fluorescent protein at the end of that gene, so that when it makes actually the protein, and attached to it is a little glowing ball. So now we know where that protein is, okay? And so basically, what happened is we were now able to take any gene, any genetic entity of interest, and we were able to instrument it so that we could see it in the cell, okay? Because that glowing ball produces at least 10,000 photons, and you can see it, that glowing ball. You can see a single protein if it is the only protein glowing, okay? If there's a field of proteins, then not. So to give you an idea, here's some early things that my colleague Tony Hyman did. Here's basically, this is the worm, and basically they were able to basically get a protein that labeled the EB1 fibers, and you can actually see the tips of the fibers growing here in the cell that's getting ready to divide. Here they basically were staining gamma tubulin. You can see the nuclear envelope disappear and the centrosomes form. And here, the double stain, so they stained one agent green, par 2, and the other one par 6. And these two genes basically establish where the head and the tail are going to be. So they establish polarity, and you can see very clearly that they've segregated to the two sides, okay? All right, and then, so what I saw though, is none of this information was being digitized, okay? And so basically some of my early work in the 2000s was to digitize this information. And so what happened is the genome didn't actually really enable what I would call systems biology. We still need to understand these systems. There's a tremendous amount to learn. But what it did allow is it allowed us to systematically, it's allowing us, even today, to systematically explore genetic systems. So for example, an early project was basically they took every protein in yeast and they did this trick of lighting it up, and they looked to see where that protein was in the cell. Another one was the Hyman and Chevry. Basically, they took every protein in the worm, and they lit it up, and they looked to see which ones were involved in cell division, and they were able to identify the 552 proteins that were critical to allowing a cell to divide. So you can see how you can get a lot of information from this. I think in the interest of time then, I'm going to skip over the next part of my talk, if I can do that. Can I manage it? You sure? Because I wanted to keep to a half hour. You sure? Okay. All right. Okay. But I was prepared to skip this because So I just gave a list of examples. And let me actually then illustrate this a little bit more carefully by talking about this project which took place over the last decade. So this is basically a fly's nervous system. So if you kind of gently dissect the fly and open it up, you can basically pull out its entire nervous system, including its spinal nervous system, okay? And here you're seeing basically the brain. This is the spine. Oh, sorry, it's animated. These are basically the eyes. The eye system has about 30,000 neurons in it. This is the core brain, which is where it makes interesting decisions, whether to eat or to have sex or to fly away, okay? And then here is the spine, which is basically about controlling the motor functions, okay, flying, walking, about 10,000 neurons. So you can see it's not a very big brain. There's about 110,000 neurons. It's a feasible number, right? Okay. So the first thing that we did is so remember, we had the genome of the fly. So because we had the genome of the fly, we were able to do the following thing, which is a little bit interesting, which is what we did is we just took out pieces of the genome, these blue pieces, and here's a protein. So we took out pieces that were in between, that were likely to be controlling whether or not this gene, this particular optamine receptor, which you would expect to be in the brain because it's a neural receptor. And we basically took out regions that look like they might be controlling the expression of this gene. And we had those basically drive a fluorescence protein. And what you see is that you see very interesting, beautiful patterns. For example, if you put this in front of fluorescence, you get just these neurons lit up, the antenna lobe. And you can see you're getting distinct regions. This is called the ellipsoid body, naturally enough, okay? But you can see the very interesting subsets. Not all the neurons are lit up, just a very small subset. And it's a little bit difficult for computer science to say, well, why is that important? It's important because it gives me reproducible access to the same neurons over and over and over again, okay? So I don't have any control over which ones are gonna get lit up. I just do it and see. But then once I've got those flies, once I've got a fly that has this construct in it, I can keep propagating that fly, and I can get back to just these neurons anytime I want to, which experimentally is very important. So what we did then is we did this. We basically we took every one of there are about 18,000 genes in the fly. We took 8,000 of the ones that we thought would reasonably be involved in the brain, the receptors, the channel proteins, and we just surveyed them. So pretty expensive business. About 4,000 of them were useless. 4,000 of them, nothing lit up, or everything lit up, right? Everything lighting up is not useful, but 3,000 of them basically gave us a beautiful covering set of every neuron. So now we had kind of a covering set of all of the things. And so then, we built a map of this thing. Let's see if the movie going, yeah. So, there was some very interesting informatics problem in terms of registering all of the images. The fly brains don't have the same shape, so we had to do diffeomorphic registering, and you can see we did a pretty good job of that aspect. Okay, but you can go even further, which is now that you've got, say, one that lights up the optic lobe, then what you can do is you can put it in front of another trick. You can put it in front of a protein that will flip under heat, but probabilistically, so it's like a coin toss. And so with a coin toss, you can decide whether to turn it on. And you can see what happened is this protein would normally light up all of these neurons in the optic lobe, and you would see basically just a big glow. But instead, it only lit up a few. And I won't explain the trick, but we got it so that it lit them up in different colors. Okay, and what you can see then is that you can actually look at these and say, yeah, these are the bodies of the neurons, and you can see the traces. So you can actually trace the neurons because of the sparsity, because they're sparse. All right, if I light up a spot with 10,000, I could take a spot that's indivisible, 2 nanometers, and if I emit 10,000 photons from that, the camera of the microscope will see it. But if I light up all of the 2 nanometer regions in a given region, the microscope will just see a huge white spot. So sparsity is critical to all of the super resolution techniques that have taken place. All right. And here's another I just include this one because I thought it was so beautiful. Okay, so it's art. Okay. Anyway, and then so we're at now, so, and I think this part of the project is still going on. I left a bit prematurely. But now basically what we're doing is we're taking this covering set, we're adding this trick of sparsely igniting it, and then we're tracing neurons. And the goal is to trace about 100,000 neurons of this brain, which has about 20,000 neurons. So in the end, we think that we will actually see every neuron on the fly brain. I wanted to talk a little bit about interdisciplinarity and in the context of things, but I think I'll just skip over it. What I want to say is that you know, when I set up the Center for Systems Biology here in Dresden, and you know, I love being here in Dresden, by the way. I'm very happy to be here and to be associated with the TUD. You guys are great. Is basically I wanted to put the cells to tissues agenda on thing, but I wanted to just make the point that, you know, we talk about interdisciplinarity, and interdisciplinarity is not really, at least I believe from my own experience, it's not just about, you know, kind of having a graduate student or a master's student running between two professors, okay? True interdisciplinary, and if you think about it, and true interdisciplinarity is going to be required for biology, okay? All right? Because biology fundamentally involves physics, it's chemistry, okay, and it's huge amounts of data, okay? So it requires basically, and very complex modeling, okay? So it's something that's going to require basically the best talents of all of the scientists, okay, in this room and all the rooms, okay, around the world. It's going to take all of us, and it's going to take all of our expertise. It's not about, right, it's not about just kind of taking a toy problem. We really have to work on these things. So I truly myself believe that the way you build interdisciplinarity is at the level of the PIs. Okay, you have to have, if you're really going to succeed and if you're really going to be strong, okay, stronger than the other competing teams, then you need to have not only the transfer between the biology, the quantitative sciences, and the technological sciences, because it takes all of those things, but you have to have that happening at all levels. You can't have it happening in a silo, and you can't have it happening in one group. There's not one of us in this room that has a brain big enough to hold it all, okay, and to know it all, okay? That's the point. So let me then talk about future trends. Do I have a little bit? Let me tell you about what I think. And again, this is strictly from my perspective and my experience, so I'm sure I'm not capturing everything that might be interesting that's going to happen here. But, you know, so I think that biology is going to become increasingly digital because of this ability to image things, okay? So I think that there are many people now that are working on basically building cellular models. So this, for example, is a little worm that has basically about 1,000 cells when it's grown up. And the idea is basically to segment those cells, okay, and build a model of it. And basically, this particular organism always develops and always has exactly the same cells. It's really pretty fascinating if you think about it. It has exactly 1,082 cells, okay? Every single worm has that many cells, except for the sex cells. The eggs are being produced. And basically, they all have exactly the same functional role. So it's, and think about it, the genome is controlling that, okay? And basically, so they all have a fixed name, okay? And you can actually literally build a digital atlas. So you can take observations of lots of worms. These cells are almost always in the same position. We understand the stochastic variability. So I can build basically a stochastic model of all of the cells. And the idea is we can layer all the information that's known about these cells onto them. Another example would be we've built a microscope that basically can image an entire mouse brain. It's basically a cubic centimeter of brain. It involves, I think, I forget, it's 100 million neurons. Okay, but what's impressive is this is a microscope that will image basically at half-micron resolution, okay, an entire mouse brain in 10 days. And that's 16 terabytes of information, by the way. And we've now actually about to deliver a microscope that does it in 2. And you can literally trace the neurons all the way through the brain. Now, this is pretty impressive. This, I think, is impressive because the neuron is 50 nanometers, but it covers, you know, 5 to 10 millimeters of distance. So it's a very fine, very long projection. We've also built this is a modern light sheet microscope, and I show it mostly because we can ask for the worm while we could build a static model for the fruit fly. This is a fruit fly developing. These are all of the cells of the fruit fly, actually the nuclei of the fruit fly. And you can see we can basically segment and track them all. And this portends the idea that we could actually build a cellular atlas of the development. In other words, we could understand what's happening to every cell through time. And so I call these atlases, and this would be all towards what I would call the idea of digital biology. I think I'll skip this one. It's about the idea that microscopes are robots, but I think you've heard enough about robots. But microscopes should be robots, and they are not. Okay, so a huge opportunity. Okay, another kind of interesting, I think, so this idea of building these atlases, of doing computer vision and being able to understand what's being seen in microscopes is, I think, very interesting. Another one is the goal of actually simulating life. Now, there have been a lot of people who wanted to do this at various levels, but let me give you an idea of how hard the problem is. So in a human being, there are 20 to 30,000 genes encoding for proteins. Those genes make about 150,000 proteins once you look at all of the variants, and the genome itself is anywhere from 1 megabase to 100 gigabase pairs. But think about an individual cell. I estimated that an individual cell has 8 billion proteins, okay? And if you're going to simulate the cell, you can't go to continuous mathematics. You can't look at these proteins in aggregate. There are some processes that occur in the cell where 30 of those 8 billion, there are only 30 of a particular kind of protein in that gamish of 8 billion. And those 30 are critically regulating some aspect of the cell, right? So it's intrinsically, it has to be a discrete stochastic simulation of 8 billion particles, okay? Now or some hierarchization of that, right? I mean, so there might be tricks to basically abstract some of it and reduce the complexity of some of it, but some parts of it you're gonna have to actually explicitly model every particle, okay? There are about 20,000 surface receptors, but there are even smaller things involved in signaling. What about simulating an organism? How many, what if you said, okay, I have a model of a cell. Well, forget about the molecules. I have a model of a cell. How many cells in a worm? 1,000. I said that already. Fly, 10 million. A human, 10 trillion. Okay, so these are for you guys, these are big numbers. Okay, but I think that we're beginning to get there. Okay, and I do think that another colleague in Dresden has recently been able to actually simulate the active 2D gel equations. There was a lot of work on simulating this. I do think that computers are getting to the point where we might actually, at some point in the near future, really be able to do this. The biggest problem we have right now is that we don't have the information we need about all the particles. I don't think we know enough to do accurate simulations, but I do think that we're going to be able to do the scale, and I do think it's just going to be a matter of time, before we're going to be able to do this. The final point that I wanted to make is there's now a resurging interest in DNA sequencing. So I said I was very disappointed that nothing new had been sequenced since the human genome, which is pretty much true. Nothing, not at any level of detail. But there are these new machines that have come out. They've been out since about 2012, so they've been out for 5-6 years now. But these machines basically produce very long reads, and they have a characteristic that they actually allow us to do near perfect de novo reconstructions. There's still a lot of work to do in actually making that happen, but it's clear it's gonna happen, okay? And so what's happened is now people have gotten interested in going out now to sequence nature. So imagine it, it's like now and the price point is actually reaching near €1,000. So I believe that in the next 4 or 5 years, for €1,000, you'll be able to look at any creature outside and say, I want the DNA sequence of that. I want the complete whole genome sequence of that, okay? Not something we could do before, but we're gonna be able to do that. There's still a lot of work to actually do that, okay, but I'm hoping that before I retire, we'll be at that state. But just to give you an idea of how much life is out there, how many different species of life are there are 1,300 bats, okay? So bats are the most common mammal. They're fascinating. They're bats that never get sick. They're bats that live 50 times longer than they should, according to their body weight. There are 66,000 vertebrates, animals with spines, okay? And there are 1.5 million known extant species. So everything that we know about is 1.5 million. The estimate is that there are actually 8.5 million things on the planet. So we don't actually know most of the things. And there are projects developing around the idea of actually sequencing all of these. So there's the BAT1K, which is the goal is to sequence all of the bats. Okay, and that's actually being driven here in Dresden, actually. There are 66,000 vertebrates. That's the Vertebrate Genome Project. And there's 1.5 million known species, the EBP. And these things are actually starting. You can go on the web and you can find a logo. And you know, like the EBP people are pretty much dreaming, okay? They're basically trying to organize it, okay? Although I did cost it out. It would cost less than 4.5 billion euros, okay? Which in inflation-adjusted, you know, currency is probably about what the Human Genome Project cost. Okay, so this is not a crazy thing. Okay, we're talking about a number that's equivalent in 2,000, you know, euros to what the Human Genome Project cost. Okay, to do all of them. There are huge logistical problems that are going to be in the way, but basically the idea is, you know, these are basically at different scales. That there's one order, actually there are two orders of bats, so the idea of the bats is to sequence all of those. The vertebrates constitute a subkingdom or subphylum of all life, and then the EBP is the entire tree of life, and the idea is to sequence these things. And geographic variation is very important. So let me conclude then by saying that I think that, and you know, the reason that I brought these three up, of course, is that this is the 50th jubileum, right, for computer science, and these problems are going to need computer science, okay? Yeah. And I hope you guys will come in. I was a little disappointed that all the kids left, okay, when the robots were over, okay, because these problems, okay, are, and I hope I point out, these problems, first of all, for your professional career, they're going to result in more citations than any robotic paper you will ever produce, okay? All right, and you will probably have more impact on mankind if you engage in one of these. For example, the outcome of sequencing all of life, the ecogenomics, is that we will learn more about our own biology and about how all of the genes in our own body function by looking at how those very same genes function across all of the clades of life, right? Because if you think about it, if you look at, you know, if you look at the difference between, you know, a person that lives to be 70 and a person that lives to be 90, that's nothing, okay? And it's like a few changes, and you're trying to see what's different about this guy from this person, and they're, you know, in a sea of 3 billion bases, and one lived, you know, 15% longer than the other. Look at bats. There are bats that live for 1 year, and there are bats that live to 50 years, and they're only 5 million years diverged. And they have exactly the same genes that we do, okay, mostly. It's almost all the same stuff. How good are my chances, okay, when looking at human beings, okay, to understand what induces longevity versus looking at bats, okay, where the range in longevity is massive, right, 50x, right? So I believe that really, if you want to understand things like cancer, immunity, longevity, okay, that you know, these are the things that I think that are really going to impact our lives in a way. I mean, I'd like my life to be easier, but honestly, I'd give anything to live longer, okay? Okay, and a robot's not going to help me live longer, okay? And so I think that, you know, and so I think that, so in a way, this is really a plea to computer science, okay, to look outwards, okay? Because I don't mean to be unhöflich, aber es ist wahr, dass informatics has tended to be inward looking, okay? I gotta say that, and I don't mean to offend anybody. The field is fascinating, the problems are fascinating, but computers don't play a role, okay, in just the internet, okay, and automation, okay? Computers are ubiquitous in science, and all of the fields, the humanities, biology, even the chemists and the physicists, although they don't know it, okay, could use us, okay? So I hope you guys, I did a little bit to maybe stimulate your thinking and get you interested in this. And again, I'm very happy to be here at your celebration. Thank you.